Dear student, today we will talk about DNA fingerprinting. DNA fingerprinting is also known as DNA profiling or DNA typing. The fingerprints are unique to each individual, even to the identical twins. Similarly, the genetic or DNA profile of each individual vary when analyzed by DNA fingerprinting. So, DNA fingerprinting is the process or a technique of obtaining genetic profile of an individual based on polymorphism of DNA. It is used as forensic technique by expert to identify an individual on the basis of DNA characteristic. This was developed by Alec Jeffrey in 1984. Now, what is DNA polymorphism? The variation of a particular DNA sequence within the individuals of the same species is called DNA polymorphism. The allelic variation are traditionally described as DNA polymorphism. For example, the gene for blue color of the flower is represented like AGGTACTT. Suppose this is sequence 1 or A. Now the variation occur at C position and C is replaced by now A. So when this is the genome, the phenotype changes to violet flower. Now, in the above case, the population of a species will have two different genes called alleles, some individual with sequence A and some individual with sequence B. So, blue and violet both flower color are visible. Now, if suppose after some time there is another variation in the same gene such that at that position now it is replaced by T. Now the color of the flower changes to white. So now we have the population with three different alleles for the same genes. So they are now known as multiple alleles. And this variation in the DNA is called polymorphism. So the variation arises due to mutation. Now, if you see the in human 99% of 99.9% of DNA is similar. Only 0.1% differs. Okay, in the individuals. So this is unique DNA that give rise to variation. The non-coding region of DNA shows more polymorphism as compared to the coding region. So the variation occurs both in coding as well as in non-coding region, and the polymorphism also occurs in both the region. We have just discussed how it occurs in the coding region. Now we will discuss about the non-coding portion. So the non-coding region is not expressed and mostly contain repetitive DNA. Okay. So what is non-coding? The one which is not transcribed and translated is called non-coding region of DNA. Now it is the region of DNA in the non-coding region in which the small stretch of DNA sequence are repeated many times. So number of repeats are there in the repetitive DNA. The same sequence or the same stretch of DNA sequence is repeated several times. That is what we mean by repetitive DNA. Now let's understand here how is this. So and you know that coding and non-coding both are present in eukaryotic cell, because prokaryotic cell will only have the gene that are expressed, that is the coding sequence. Okay. So eukaryotic cell, the DNA is present in mitochondria, chloroplast and nucleus, and this total DNA is called genomic DNA. Okay. Now which consist of coding as well as non-coding region, and non-coding consists of repetitive DNA, whereas coding region codes for a gene. Now this genomic DNA is, when subjected to density gradient centrifugation, is separated into two different bands. One of the band is called satellite DNA, which is of non-coding DNA. Now, why it is known as satellite DNA? Due to repetition of short DNA sequence, they tend to produce different frequency of ATGC, that is adenine, thymine, guanosine and cytosine, thus have different density from the bulk genomic DNA and form second or satellite band. Okay, and these satellite DNA usually they have a repeats which are called tandem repeats. Now what are tandem repeats? They are the short DNA length repeated multiple time in the genome and which are adjacent to each other. So satellite DNA consists of repetitive DNA which repeat in the tandem way, and it contains the variation in the DNA. Based on this variation, it is again, it is means the satellite DNA is classified into different categories: macrosatellite, microsatellite and mini satellite. Now basically the difference is in the number of repeat unit and how much times it is repeating. So we have macrosatellite short repeat, microsatellite 2 to 6 base pair repeat and mini 10 to 100 base pair repeats. These mini satellite and macrosatellite, they are also known as VNTR, that is variable number of tandem repeats. Now this macro satellite repeats for 1000 times. The number of repeat is 1000 times, whereas the mini satellite is 4 to 10 times, and mainly they occur um, in the centromere and telomere region, whereas microsatellite they are distributed throughout the genome, and they are consist of two six base pair repeats. They are also known as simple sequence repeats or short tandem repeats, and the number of repeats varies 8 to 200 times. Okay, now these repeats are important in DNA fingerprinting. The repetitive fragment of DNA present in multiple copies in genome, and these sequences exhibit a high degree of polymorphism due to variation in the number of their repeat unit. Okay, so let's also understand what is repetitive DNA. Repetitive DNA is divided as satellite DNA and interspersed repeat. Now satellite DNA is separated on the basis of density gradient centrifugation from the genomic DNA, and basically it consists of tandem repetitive sequence. Okay, now if you consider short tandem repeats and VNTR, that is variable number of tandem repeats which are kind of satellite DNA, they form the basis of DNA fingerprinting. And the basic difference is the short simple repeats or short tandem repeats is short repeat as compared to VNTR. Okay, for example, if you see over here, to understand the repeat AG, 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 AG. So AG forms one unit of two base pair and it is repeated 4 times. So the number of times it is repeated is 4 times. Similarly, if you see AGG, so AGG forms one unit and repeated here 5 times. So number of repeat is 5 times and unit of repeat is 3 base pair. Similarly, one unit consists of 7 base pair and repeated 3 times. So the number of repeat vary from person to person. Therefore, there is variation in the length of repeat and also in the sequence of different individual. So the variation arises because of the length of repeat, or you can say number of times it get repeated, and also sometimes there is variation in the sequence also. So for this reason the repeats are known as variable number of tandem repeats. So the repeats they vary from person to person in a population, in the human population if you talk. Now both short tandem repeat and VNTR are co-dominantly inherited. This is the property. Therefore, an offspring will inherit repeat pattern from both the parents. Therefore, these repeats are used in the detection of the real parents. The STR and VNTR are flanked by short conserved sequence, which is known as consensus sequence, that are used to develop primer during the PCR. So they are also used as probe when they are radio labeled. VNTR are the source of RFLP, that is restriction fragment length polymorphism, during DNA fingerprinting and linkage analysis that is based on physical map. So there are also source for RFLP. Now what is RFLP? Restriction fragment length polymorphism is a technique used to analyze the difference in VNTR. Okay, so variable number of tandem repeats are there, and how you will identify that these are present in different individual, what is the pattern, so that is done by RFLP. So RFLP is used as technique, okay, to analyze the difference in VNTR in different individual, and it is based on the fact that DNA molecule have unique site where restriction enzyme cleaves the DNA molecule and generate the fragment. The fragments are separated by gel electrophoresis based on molecular weight. The smaller fragment of digested DNA molecule move farther on agarose gel as compared to the larger fragment. The process produces a pattern of DNA restriction fragment that is unique to all people. Okay, so we'll see, let's understand how this happens. uh, there is person 1. So we have taken the homologous, the DNA from the homologous position and of person 2. So now there exists a variation in 1 and 2 as far as the repeats are concerned. So same repeat with little variation. Now we have used the enzyme EcoRI. So if you see, first person will have the site at specific place, whereas the second person, because of the variation, will have uh, 2 or 3 different, you can say, sites of EcoRI. So the first one generate 2 fragment, whereas the second one generate 3 or 4 fragments. Okay, so when you do the or when you run them, this fragmented DNA on the gel electrophoresis, you will see the pattern. For the first one, you will see, since they are large pieces, so they will be toward the upper end from where we run the gel. Okay, it is always from negative to positive, so run in this direction. And the second, you will see 3 different band are seen. So with the same enzyme, different length of DNA fragments are generated. Okay, different lengths are generated, and this is called RFLP, that is restriction fragment length polymorphism, which is also categorized under DNA polymorphism. So DNA polymorphism, you can say, is a term which includes basically the variation in the length of DNA because of number of reasons. In the above case, there is one base pair differ in second person for the repeat as compared to the first person. RFLP fragment are also known as genetic markers or DNA marker. They are also known as molecular marker in plant breeding experiment, and they are used to tag the desirable gene. Now this desirable gene is with a known trait and unknown location. So RFLP, these, you can say, the polymorphism is also utilized as the genetic markers. Okay. Now we will see the DNA fingerprinting principle. Now since you know now what is polymorphism and how they are useful, we will discuss now the principle of DNA fingerprinting. It is based on the principle that each individual has unique pattern of tandem repeats in their DNA that could be recognized by performing restriction fragment length polymorphism. So whatever is the difference in the, or you can say variation in the tandem repeats, how you make sure that there is, or how you will analyze, the analysis is done by RFLP. Okay, so this is what is DNA fingerprinting principle. Now we will see the technique how it occurs. So first is you will isolate the DNA from the cell. The cell may be skin, hair, blood cells. Okay, now to isolate the DNA, we just get rid of the protein part, lipid part, RNA part. Okay, and then you do the centrifugation, so the other thing is separated from the DNA, and then DNA is precipitated with alcohol, and we check the purity of DNA by running it on the agarose gel electrophoresis. Once we make sure that we have the pure DNA, it is amplified by using PCR, polymerase chain reaction, and we get several copies of DNA. Now this DNA is digested by restriction endonuclease, and these restriction endonuclease, they cut the DNA at specific site, okay, and produces the fragment, and these fragments are run on the electrophoresis agarose gel. So depending on the molecular weight and the porosity of gel, they are separated. The larger fragments will be toward the negative electrode, and the smaller one will be toward the uh, positive electrode, as the DNA run from negative to positive terminal during the electrophoresis. Now the fragment on the gel are subjected to denaturation. Now once you have this fragment, so in the denaturation, double stranded is converted into single stranded DNA, and then they are transferred to the nitrocellulose or nylon filter. So now we have all the DNA on the filter instead of gel. Now this is called southern blotting, and in the southern blotting the DNA is transferred to the membrane, and then it is hybridized with another DNA. So the DNA fragment are now subjected to hybridization with the complementary DNA with the VNTR probes. Okay, now whenever we are using VNTR as probes, these probes are actually radio labeled, so that we can detect the radioactivity and we can detect the pattern of bands. So the probes are radio labeled with short pieces of single strand complementary DNA that bind to the DNA fragment of interest. So you can see the probe hybridization is done over here with the probe, and then you can see the hybridization is done, and now again the single stranded DNA is converted to double just because of the hybridization. So the detection of hybridized DNA is done by autoradiography, in which X-rays are used, and you will see the band pattern on nitrocellulose filter membrane. So this is the technique. Now how this technique is applied, uh, we'll just see the two application. First is to identify the correct pattern. So here suppose this is the final you can say band pattern that is obtained on the nitrocellulose membrane. So this child, okay, belong to male 1 or male 2. So which one is the father of the child? So then we have to compare. So over here you can compare that most of the band pattern are similar with the male one. See, with the mother, so many uh, patterns are similar, so the child belongs to the mother, but who is the father? So this is how it is detected, whereas male 2, not much similarity is there, so the male one is the father of the child. Similarly, when there is crime scene, some murder or something like that, then what we do, we run the DNA fingerprinting of the specimen, victim, and whatsoever is the number of suspects are there. Okay, so The victim is their specimen, which is again the standard, you can say, DNA, and suspect 1 and suspect 2. So here you can see the most of the band pattern are similar with suspect 2. So the DNA is taken from the blood stain at the place of crime and matches with that of suspect 1. So this is how it is identified that suspect 2, not with 1, but suspect 2. So the banding pattern matches with suspect 2. So this one is the criminal. So this is how the DNA fingerprinting based on the band pattern help to identify the person. And this band pattern actually come from the RFLP, which is done, and the RFLP actually identifies the variations in the tandemly repeat DNA, and the tandemly repeat DNA are STR, SSR, and VNTR, and these are nothing but they are satellite DNA, and the satellite DNA is nothing but it is separated from the genomic DNA. So basically the DNA fingerprinting um, is actually based on the DNA, and this has different unique pattern of banding. Okay, so this is all about DNA fingerprinting. Thank you.